So welcome everyone to the first Anatomy tutoring session. We are very excited to have you all here. We're very excited to host our second competition. Before we get started, we just have a few announcements. First and foremost, I wanted to just introduce myself. My name is Dr. Jenna Haggerty. I am one of the co-founders of Anato-Bee, and I work at Thomas Jefferson University in Philadelphia. And I teach anatomy to medical students and physician assistant students. Teaching all of you and providing this opportunity is near and dear to my heart. It's very important to us that we provide opportunities to people at a high school level so that they can get into a job and enjoy what they do just like we do. So I'm going to ask Doctors Piscira and Peterson to introduce themselves. They're the other two co-founders. And I'll hand off to Dr. Piscira. Go ahead and introduce yourself.
Hi, Dr. Haggerty. Thanks for that. So I'm Dr. Piscira. I'm a faculty member at the Edward Via College of Osteopathic Medicine in Auburn, Alabama. I've been loving the journey here and in the sunny weather down here, but really excited to reach all of you guys, no matter where you're hopping on this Zoom call from. So really, really glad that you're here and really excited to spend more time with you over the next couple of months as we prepare for the competition. All right, Dr. Peterson, you're up.
All right. Hi, everyone. My name is Dana Peterson. I am a full professor at Drexel University College of Medicine. I have been teaching the anatomical sciences for a really long time, but like Dr. Haggerty and Dr. Priscilla said, what keeps us motivated and inspired is being able to teach it and freshly to high school students that are learning all of this content for the very first time. I'm going to ask, and I hope I'm not catching Dr. Priscilla off guard, but we would love to have you fill out our Anato-Bee survey if you haven't already done that. It's a motivational survey, and maybe she could put, maybe not right now, but we'll get it up in the chat. And then the other thing I would highly, highly, highly encourage you to do is please recruit one or two other students to join us. We're really trying to reach as many students across the United States as we possibly can. And I do see some familiar names here. So I'm so glad you're back, returning with us for a second year of competition. Great to see you, and it's going to be a great session. Okay, that's it.
I want to take the opportunity to introduce Dr. Bill Frank. He's a supporting member of the Anatomy, and he's going to be our moderator for all of these tutoring sessions. So Dr. Frank, do you want to say a few things about yourself?
Okay, welcome everyone, and welcome to year number 2. I wish you all the best of luck and success over this next year. So I will be your humble moderator for these tutorial sessions. To give you a little bit of background on me, I served for 23 years in private practice as a doctor of chiropractic, and about 11 years ago came into academia. So currently I am an associate professor at in the Department of Neurobiology and Anatomy at the Drexel University College of Medicine. When I'm not teaching in the anatomy lab, or in classroom, I love spending time with my wife, Miriam, and listening to or playing music from Mozart to Megadeth. So I live it. I live by the mantra, anatomy is like a box of chocolates. You never know what you're going to get. So with that, I would like to introduce our first presenter.
I do have a few other announcements. I'm so sorry, Dr. Frank.
No problem.
But we are very excited to announce the team of medical students who will be tutoring you this afternoon. We do usually jump right in and get started, but I want to make sure that we get through the few announcements, being this is our first tutoring session. So you guys all know why you're here. We know Anatomy is a national competition. We are going to be assessing you in the anatomical sciences. That includes anatomy, histology, and embryology. You're obviously here because you're familiar with our website. I just want to say a friendly reminder, we do provide free study resources for you, including these tutoring sessions. So make sure you go to the website and check out all the learning resources that's provided. This will include the learning objectives that you will be using to compete in the competitions themselves. If you haven't registered for the competition already, please do so. You will register individually. This will not be a registration with your school. Let me see here. So, and in addition, Dr. Pesquera did put the research survey up. I'm also, so this is all at the beginning of our chat. I'm going to put the link to register. So we'll have a couple of key points here that you can refer back to within this recording, but you can also go to the website and find the registration link. We just want to make sure that all of you are registering. And like Dr. Peterson said, share with your friends, share with your high school teachers. You can share our information, share our website. We had about over 700 registrants last year for the local competition, and we want to keep those numbers growing. So for the local competition, that is what we're tutoring for currently. So we will be going over these local learning objectives only. And we are going to be preparing for that competition that is going to be held on Saturday, January 25th in 2025. This is information you can find online, but just so we're all on the same page, it's our first session. I just want to go over these key points. So when the local competition, January 25th of 2025, and then those top placing students will be invited to come to the in-person regional competition, which will be in March 8th and 9th, 2025. All of this is to get a chance to win a financial award to attend a summer experience in STEM. And we are currently recruiting host institutions for that regional competition. And when we find out more about that, we are going to be more than happy to share that with all of you. Tutoring agenda basically goes like this for those of you who are new here. We start at 6 p.m. and we typically go till 8 p.m. at the latest. We start with some brief announcements and then we hand it off to Dr. Frank, our moderator, who will be introducing our wonderful second-year medical students who will be tutoring all of you. We'll start with anatomy and then histology and then embryology. The sessions will take 30 minutes each or less. And then we will give 10 minutes for you all to ask questions, whether you unmute yourself or you ask them in the chat. So with that, that's all that I had. Again, just a reminder, this is being recorded. It will be put on our YouTube channel. So if you do not consent to being recorded, please keep your camera and your microphone off. Before I hand it off, did we have anything that we wanted to mention before we get started? All right. Okay. So all of you are wonderful. We're excited. And now I'm handing it off to Dr. Frank.
Okay. Sorry for jumping the gun there a little bit. Okay. Our first presenter this evening is Hailey Long. Hailey Long is from Madison, Alabama, and graduated from the University of Mississippi in 2023 with a BS in biological science and a BA in psychology. She is currently a second-year medical student at Edward Via College of Osteopathic Medicine in Auburn. When not studying, Hailey loves reading and spending time with her sweet cat, Bobbin. Maybe Bobbin will make an appearance this evening. Hailey's favorite part of anatomy is musculoskeletal anatomy. So with that, I open this up for Hailey. You can put your slides up and begin your presentation. This will be the anatomy presentation for the evening.
Thank you for that introduction, Dr. Frank. Oh, this is the right one. Okay. Let's see. Okay. Good evening, everybody. This is the Introduction to Anatomy presentation. I would like to acknowledge that this presentation was adapted from, that was the cat. Thank you, everybody. We'll back up to the beginning. This was adapted from a previous presentation by Hannah Lufkin, now a OMS 3 student at VCOM-Auburn. Okay, first, our learning objectives for today. We're going to talk about the 12 organ systems and their primary functions. We're going to talk about anatomical position and then how to describe the body directions, regions, planes, and movements. We're going to look at the major body cavities and figure out what organs are in each, and then define homeostasis and how it's maintained by negative and positive feedback. So first, our organ systems. We have 12 total organ systems that are all pictured on this slide, and we're going to break them down each individually. First, we have our integumentary system, which encloses our internal body structures and is the site of many of our sensory receptors. This includes our hair, our skin, as well as our nails. And it is the largest organ in your body. Here is our skeletal system, which we're probably all most familiar with. It supports our body and enables movement in combination with our muscular system. It includes cartilage, bones, and joints. And then we have our muscular system, which enables movement in combination with the skeletal system and helps to maintain our body temperature. It includes our skeletal muscles as well as our tendons. Next, we have our nervous system, which is used to detect and process various sensory information, and it also activates bodily responses. This includes our brain, our spinal cord, as well as our peripheral nerves. Next, we have our endocrine system, which secretes various hormones throughout the body and helps to regulate our bodily processes. Some of the organs involved with this include our pituitary gland, our thyroid gland, our pancreas, the adrenal glands, the testes, and the ovaries. Next, we have our cardiovascular system, which delivers oxygen and nutrients to tissues via the blood, and it equalizes our temperature in the body. It includes our heart and our various blood vessels. Next is our lymphatic system, which is used to return fluid to the blood. It also helps to defend against our pathogens. And some of the organs involved include our thymus, lymph nodes, spleen, and the lymphatic vessels. Next, we have our respiratory system, which removes carbon dioxide from the body in exchange for oxygen and then delivers that oxygen to the blood. It involves our nasal passage, the trachea, and the lungs. Next is our digestive system. It processes food for use by the body and then removes waste from undigested food from the body. The organs involved here include our stomach, liver, gallbladder, large intestine, and small intestine. Next is our urinary system, which helps to control the water balance in the body, and it removes waste from the blood and excretes them via urine. It includes our kidneys and the urinary bladder. Next, we're going to talk about the reproductive systems. There are two different ones. We have testosterone-driven anatomy, which is developed by, caused to develop by the hormone testosterone, and it produces various hormones and gametes called sperm. And then the system also enables delivery of sperm to the egg for reproduction. It includes our epididymis and the testes. Then we have our estrogen-driven anatomy, which is caused by development of the hormone estrogen, excuse me. It produces hormones and gametes called ovum, and then supports our embryo and fetus until birth, and then also produces milk for the infant via the mammary glands. It includes our ovaries and the uterus. Next, we're just going to talk about anatomical position, which is pictured here. Anatomists like to have a standard way of viewing the body, and that's called anatomical position. And this is helpful in communicating where things are on the body and just having a standard language. So anatomical position specifically involves a person standing upright with their feet shoulder width apart and parallel, and then with the toes pointing forward, and the arms held out to the side with the palms facing forward. So then, no matter how the body is oriented, people can talk about the body in anatomical position, and we know exactly where that person is talking about. And anatomical position is important for understanding body direction and how we describe that. So our first body direction is left versus right, which hopefully we all know, but it's very easy to get confused. And especially when you're looking at a patient, it's important to remember that when you are face-to-face with a person, your right hand is on that patient's left side. And that is something you need to remind yourself of when you're looking at graphs or images and things like that. Next, we have superior versus inferior. Superior or cranial means above or toward the head. So in this picture, we have superior up at the top, inferior at the bottom. And inferior, or sometimes referred to as caudal, means below or towards the tail. Next, we have anterior versus posterior. Anterior or ventral means towards the front of the body, while posterior or dorsal means towards the back of the body. Next, we have lateral versus medial. Lateral means towards the side of the body, while medial means towards the middle of the body. As we can see in our image on the right, lateral is pointing outwards towards the arms, while medial is positioned at around the belly button area. Next is proximal versus distal. Proximal means closer to the origin or center of the body, while distal means further away from the origin or center of the body. So in our image here, we have distal being the palm of the hands, while proximal is closer to the shoulder in your upper arm region. Then we have deep versus superficial. Deep refers to further from the surface of the body, while superficial is closer to the surface of the body. So in this image, superficial is labeling more the skin or the skull, and then deep is referring to the brain on the inside of the skull. Now we are going to do a poll, and if Dr. Pescura could launch that poll, I would appreciate that. Our first question is going to be, the brain is blank to the skull. A, posterior, B, deep, C, distal, or D, lateral. We're just going to wait a few seconds for people to answer and then talk about it.
You guys are doing really good so far. We got about 66% of you guys participating in this. I'll just give a little bit longer. All right, and we're at about 86%. So I'm going to say, get all your answers in 5, 4, 3, 2, 1. And I've shared this.
Okay, the correct answer here is in fact B, which about 69% of you answered correctly. Let's see, can we get this to move? Yes, there we go. So the brain is deep to the skull, and we can go back to the other picture if we would like. Let's see. So we're looking at this picture down here. So the brain is surrounded by the skull, and so we could say that the skull is completely superficial to the brain, while the brain then would have to be deep to the skull. The brain is not posterior to the skull because there are parts of the skull that are posterior to the brain. And I think the other answer was distal, which I'm not sure of a good way to explain why that one is not making sense to me, but I hope that explanation made sense from the picture. So our next question is, the inferior portion of your face itches. What structures would you scratch to relieve the itch? And if we could have the second part of the poem, thank you. So A, your eyelid, your B, your temple, C, the forehead, or D, the chin. We'll wait a few more seconds for people to answer.
All right, you guys are doing good with this one. I'm going to go ahead and close the poll in 5, 4, 3, 2, 1.
Okay, the consensus is right that the answer here is D, the chin. And that's because the chin is the most inferior part, the most inferior structure that's mentioned in our answer choices here. Your eyelids, I'm not sure how best to describe, but they're more medial on your face, and more on the superior half of your face, while your temples are out here very laterally, so we would describe them as more lateral. Your forehead is very superior, and then your chin is the most inferior portion of your face.
And Hailey, I think you're doing a wonderful job. I just wanted to point out, and maybe you can elaborate a little more, that these directionalities can be zeroed in on a certain area. So even though Hailey said superiors towards the head and inferiors towards the feet, if she says, what part of my face, your face can be also segregated into superior, inferior.
Yes, thank you so much, Dr. Haggerty. That's a great point. So yes, we can focus in on different parts of the body when we're talking about these terms, and I think that's the perfect way to describe it, because it's easy to forget about that. Okay, next we're going to talk about our body planes, and this is the picture that comes with it. It's a little confusing, but we're going to get through it. First, we have our sagittal plane, which divides your body into right and left halves, straight down like the midline of your body. And the mid-sagittal or median plane is down the middle of your body. So let's specify here that sagittal can be not necessarily centered, or I guess median plane or mid-sagittal plane is being very specific that the sagittal plane is down the midline of the body, like cutting through your nose, or I guess sagittal plane could be a little bit off-centered, perhaps.
Right, and I'll just jump in, Hailey, it's
Yes,
Dr.
ma'am.
Peterson. So again, we can use sagittal, and that means that not necessarily are we dividing them into equal right and left halves. So sometimes that's designated as parasagittal. The prefix para means close to or next to, but not necessarily on the midline. And then, like Dr. Haggerty suggested, we could look at, for instance, the upper extremity, and we could talk about dividing the upper extremity sagittally or in these other planes that you're going to speak about. So it doesn't have to be the full body. It can be a body part. You're doing great, Hailey.
Thank you. Okay. Next is your frontal or coronal plane, which divides the body into anterior and posterior. Because it can also be called the coronal plane, I like to think of it as the crown, because it can divide your body in like a front half and then a back half. And then next we have our transverse plane, which divides into upper and lower sections. And it's often illustrated where it's cutting the body into an upper torso and then your lower extremities. But as Dr. Peterson and Dr. Haggerty were mentioning, the transverse plane can be applied to various body parts as well, to be more specific for a general region. Next, we're going to talk about body movements, and these occur around the planes we were talking about. So first we have flexion, which is anterior bending, and then extension, which is posterior bending or straightening from a flexed position. So our first graphic here is showing flexion as flexing the head forward and then extending the head back. And we can also do this with the entire torso, where we flex forward or we extend backwards. But flexion and extension can also apply specifically to our extremities. And when we're talking about our limbs, flexion is usually decreasing the angle between bones, while extension is increasing the angle and straightening the joint. So in our image, Flexion of the shoulder is specifically bringing the arm upwards, while extension is bringing it down and then potentially back behind you. And then with your lower extremity, extension is straightening out that knee joint, while flexion is bending your leg backwards. Then we can talk about abduction, which moves the limb laterally away from the midline of the body, and adduction, which moves the limb toward midline or across the body. And I know that I like to think of the difference between these two as adduction is adding back to the body. So you're bringing your limb back towards your torso, or towards, like you're bringing one leg back towards the other. So abduction and adduction can apply to your upper extremities, but also your lower extremities. Then we have circumduction, which is movement of a body region in a circular motion while one end stays stationary. And we can think of this happening at the shoulder, where we can circumduct our arm, and that's like making little arm circles, but your shoulder is not actually moving, it's the lower part of your arm in relation. And then we have rotation, which is more of a twisting movement, and we can have different types of rotation. You can rotate your head, but you can also with your I know at least legs is shown in this picture. You can laterally rotate your leg out away from your body, or you can turn your leg medially, and that's called medial rotation. Next, we're going to talk about pronation and supination. These specific terms apply to movements of the forearm. So supination moves your palm side of the hand upwards, and that's into our anatomical position. And then we have pronation, which moves the palm side of the hand backwards or down, just out of that supination form. So we have our supination, and then we have pronation. Next, we have
one thing I want to clarify really quick, Hailey.
Yes, ma'am.
Supination can also just refer to the general position anatomically. So typically when you
Yes.
come into the anatomy lab, you guys, if you come to regional competitions, we'll get an opportunity for that. That will say that they're in the supine position. If you see that they are in an anatomical position with their faces, like they're laying down on a bed, versus they could be prone, in which case we could be looking at more of the features of their back, for example, and then we'll say that we need to look at the body in prone position with, again, their face facing downwards, like they're laying on their tummies. But doing great, Hailey.
Great addition, Dr. Briskin. Then we have our movements of the ankle. We have dorsiflexion, which lifts the foot and the toes upwards. And then we have plantar flexion, which points the toes downwards. And I think that image there really helps to clarify that. They both have flexion in the name, which I think is the most confusing part. But the plantar surface of your foot is the bottom of your foot. So when you're plantar flexing, you can think that you're moving the bottom of your foot downwards. Then we have inversion, which turns the bottom of your foot towards the midline. So you're turning it medially. And then we have eversion, which turns the bottom of the foot away from the midline, so laterally. Next, we have movements of the jaw or the shoulder blade. Here we only have images to demonstrate these terms for the jaw, but they do apply to the shoulder blade. We have protraction, which moves the jaw or shoulder blade forward or anterior. We have retraction, which moves the jaw or shoulder backwards or posterior. And we have elevation, moving it upwards, or depression, moving the jaw or shoulder blade downwards. And I'm thinking lastly, we have opposition, which is just bringing the thumb to the tips of the fingers. And that can be any of the fingers. So now we're going to do another poll. If Dr. Piscura could please launch that. Our first question is, when you want to shake someone's hand, your arm goes into blank at the shoulder. So think back to those images and descriptions. I can see this one is a tough one. It's taken a little while. And this is a confusing question.
Feel free to do the motion
Oh, yes.
anytime you're in a test
shoulder.
or anything. Yeah.
Yeah, I'm going to shake your hand. So I'm lifting my arm like this.
Oh, here
If
you
that
go.
changes your answer.
All
I
right,
don't
we
know if
got
that's matching up on anybody's screen, but I'll shake your hand, Hailey.
Oh, yeah. Oh, yep. You were even doing it in the right direction.
all right. I'm going to close this poll in 5, 4, 3, 2, 1.
Okay. So the correct, oh Lord. The correct answer here is flexion. And this is confusing. I remember when I was making this slide, I was like, oh yeah, that is flexion. So there's no reason to be concerned that you didn't get this answer right if you did not. So flexion at the shoulder is coming up anterior, like you're moving your arm in front of yourself. We talked about extension is more pushing your arm back behind you when we're talking about the shoulder, and extension is coming out in front of you. I hope that made sense. If it does not, please ask in the chat or feel free to speak up if you need to. Our next question, if we could launch the second poll, is what movement would you have to do to hold a bowl in your hands?
All right, I'm going to give about 5 more seconds for this one. So 5, 4, 3, 2, 1.
Okay, so most of you answered A, which is our correct answer. Holding a bowl, you have to do this, and that's how I like to remember supination, is you have to hold a bowl of soup in your hands to supinate. This would be pronation. Abduction is coming back to your body. Let me see if I can stand up. So if my arms are out like this, and then I do this with my arms, that's abduction. No, excuse me, I've said that wrong. This is a deduction. This is a abduction. A deduction, abduction. The words are very similar, so that's why people like to also emphasize the first part of the word, AB or AD, to clarify what they're talking about. Protraction is referring specifically to your jaw or your shoulder blade, and that's pushing it forward. Like it's not a cute movement, but it's like that. And then dorsiflexion is specific to your foot, and that's moving your foot upwards. This is my hands, but it should be for your feet. Okay, next, we're going to talk about our body cavities. In our body cavities, we have a ventral or anterior body cavity that contains our thoracic and abdominopelvic cavities. Our thoracic cavity contains our lungs and our heart. And then our abdominal cavity contains different digestive organs like your stomach, liver, pancreas, gallbladder, small intestine, and large intestine. And then we have our pelvic cavity, which contains the urinary organs, reproductive organs, and then the last bit of your digestive organs there. Our diaphragm is the muscle that separates the thoracic and abdominal cavity. So our diaphragm would be the most inferior portion of our thoracic cavity and the most superior portion of our abdominal cavity. And there is no true anatomical separation between our abdominal and our pelvic cavities. So it's just kind of a cavity that we made separate from each other without a real structure in between them. Next, we have our dorsal or posterior body cavity, which contains the cranial and vertebral cavities. The cranial cavity contains our brain within the skull, and our vertebral or spinal cavity contains our spinal cord within the vertebral canal. That one went a little fast, so let me know if there were any questions there. Next, we have homeostasis. Homeostasis is a physiological process in which the cells throughout the body attempt to maintain internal conditions within a narrow range. So, for example, our body temperature and our blood pressure. Each of these conditions has a set point in a normal range. So we know that our set point for body temperature is somewhere around 98.6 degrees Fahrenheit, but the normal range can be between 97 and 99 degrees. But we know if someone has a temperature of 101, that's out of normal range and is not healthy to be maintained for long periods of time. Or if somebody's body temperature was 94, that's also concerning because it's outside of our normal range. Homeostasis is maintained via two different feedback mechanisms, negative feedback and positive feedback. And now we're going to talk about those two specifically. So negative feedback reverses a deviation from homeostasis back to the normal range. And there are three components of a negative feedback system. We first have our sensor, which detects the change in the physiological value. The value is then reported to a control center, which will then activate an effector. And the effector will then cause a change to bring the value back to normal. And negative feedback occurs, as we mentioned, with body temperature and blood pressure. So if our body temperature exceeds 37 degrees, the nerve cells in the skin and the brain will then sense that change. That will be reported then to a temperature regulatory center in the brain, and then that regulatory center will activate an effector, your sweat glands, which will then release sweat to attempt to cool down the body. Positive feedback intensifies a deviation from homeostasis until a specific endpoint is reached. And the most common example used is childbirth, which is shown here. Positive feedback is a much less common mechanism for homeostasis than negative feedback, but it is still important for maintaining homeostasis. And here are our references. You can access any of these textbooks, and to look up more information, they are free and open to anyone to use. And if we have any questions, we can take those now.
Thank you, Hailey. I thought you did a great job. I know that a lot of those terminologies can be something that you need to sit with. So it's really okay if some of that was going over your head a bit, or going superior to your head if we're using anatomical terms. But I promise if you spend some time with it. Yes, exactly. But great job, Hailey. We appreciate that.
Thank you, Dr. Briskin.
Okay. If you do not wish to open your mic and ask a question, you can certainly do so in the chat. We'd be happy to answer it. And give it a minute here. There are no, oh, we do have one. Juan, do you want to ask your question?
Yeah, I was not entirely sure about the question about the palm that we
Okay.
have in the pool. So is supination necessarily a movement, or is it rather a position in which we look at the hand?
Like Dr. Pascura mentioned, the word can be used to describe both. Supination, I think, is more specific to the action of putting your palms upward while pronation is down, but a person can also be supine, which is like laying in basically anatomical position with your palms out. So that's also how that term can be used. Thank you for your question. Did that answer your question?
Yeah, thank you.
Okay, thank you.
It's a good question too.
Yes, excellent question.
The Asian at the end kind of is the difference. And the other thing I just wanted to put out there is that if anyone also wants to ask Hailey about her experience as a medical student, kind of any questions that you have for our presenters are all free game. So
I have a question for the dorsal cavity. Could I know the difference between cranial and vertebral again?
Of course. Let's go back to it. I think the pictures help. Oh, this is right. Okay. So, and if any of the more learned people would like to chime in, cranial cavity, your spinal cord is continuous with your brain. So I think this is another one where the difference between them is more for our understanding, but also the bony surroundings changes. So that's, I think, also why the name changes. So cranial cavity is entirely limited to your cranium, the actual skull, and our vertebral cavity starts once we're now in our vertebral canal surrounded by the bony vertebrae of the spine.
And then
Okay,
I'll,
thank you.
I will say too, so we haven't gotten to the embryology yet, but you'll hear some anatomist, and I don't think we have anyone here that does this regularly, but I have a supervisor that does this all the time. So you'll hear us say anterior or posterior. So usually anterior is my face and posterior would be the back of my head. But sometimes they will say dorsally, which is basically saying the same thing as posterior, back of your head, or ventrally, which is my face. And this is really from embryological development. We don't get too much into neurodevelopment, but that's how the brain will develop from a dorsal ventral position. And then when we talk about neuroanatomy, we often refer to the positioning of the brain and the spinal cord as dorsal ventral. Again, all from development from embryology. So that dorsal body cavity, that's where that dorsal name is coming from. But we can also see and appreciate that, like the spinal cord, it's positioned posteriorly, and it's going to be contained within what's called the vertebral cavity. So that's a little bit more in-depth knowledge as to why you're seeing this word dorsal and maybe not posterior.
that's a really good question too. Other thing that I'll add is that there is continuity between these cavities, like Hailey had mentioned, and same thing with the anterior cavities as well. For example, the abdominopelvic cavity, you could call it the abdominal or the pelvic cavity, but because they're continuous with each other, sometimes they're just called the abdominopelvic cavity.
Thank you.
All right, great questions. I'm going to say for the sake of time, if anyone wants to keep putting questions in the chat, they can. But Dr. Frank, would you keep the ball rolling for us?
Absolutely, be happy to. So we will move from anatomy to histology. So we have with us Andrew Briskin, who's from Bryn Mawr, Pennsylvania, and graduated from the University of Pennsylvania in 2021, majoring in health and societies with a minor in chemistry. He is currently a second-year medical student at the Sidney Kimmel Medical College at Thomas Jefferson University, with an interest in surgery. In his free time, Andrew enjoys climbing, hiking, golf, and watching movies. So with that, Andrew, you can take charge here.
All right, thank you, Dr. Frank. Let's start sharing the screen real quick. Okay, so today we are going to be talking a bit about epithelium, glands, and some stuff about connective tissue. So to get right into it, these are basically just learning objectives. So we're going to start with the epithelium and glands, just to give a quick rundown of what I'm going to talk about. So we're going to go into composition and function of the basement membrane and what exactly that means and what it is. We'll get into that briefly. Also, the morphological differences between types of epithelium. And so that's really talking about like what different types look like in terms of, number of cell layers we might see, the shape of the cells, and what exactly that means in terms of their function, which is sort of this third one, just talking about like different types of epithelium and then what they're doing exactly based on their appearance and what we know about them. And then lastly, we'll talk a little bit about glands, and just structural and functional differences, and what exocrine versus endocrine glands mean and what exactly each of those do. So there we go. Okay, so to kind of, start at the very beginning, I'm sure some of you have had a little bit of background in this in terms of, what you've seen in biology classes or whatever. But to talk about just the anatomy of a cell, specifically when we're talking about epithelium, we are going to look at it in terms of one end. There's two ends of the cell. So we have an apical end, and that's going to be sort of our top end, which is going to be an opening or going to be faced towards an opening or a surface or what we call a lumen. And a lot of times those will have specialized structures, which we're also going to get into a little bit later. On this diagram here, we have some cilia. We'll get into what that means. And then on the basal end, that's going to be sort of the bottom of the cell. And that's going to attach to what is a basement membrane. And then just briefly to kind of talk about cellular contents, we have our nucleus in the middle, and that's going to contain the DNA of the cell. Cytoplasm, which is that sort of jelly-like substance that's going to surround the nucleus and take up a lot of the space in the cell, and then filled with organelles, which are going to perform a variety of functions. I'm sure some of you have heard of much of these: mitochondria, endoplasmic reticulum, Golgi, etc. So to get into what we're going to talk about today. So with our last presentation, we heard a little bit about macroscopic stuff. Now we're going under the microscope. So what histology is, I honestly didn't know what that word meant till I started medical school. So basically what it means is study of microscopic structures. So we're looking under the microscope at cells specifically. And like we just talked about with the sort of architecture and structure of the cell, we have a nucleus, we're going to have a cytoplasm and plasma membrane. But the thing is, cells are mostly made of water. So a lot of times when you look at them, if you just take like a little cheek swab or something and you put it under the microscope, you're probably not going to see a whole lot. And so what we do to look a little bit more in depth at cells is we're going to stain them. And when we stain cells, essentially what happens is we can see the different elements of those cells. And so let's see if I can annotate here. But, basically what we're kind of looking at is when we stain them, we can see like an outer membrane like here. And then we can also see like this inner part, and that's going to be a nucleus. And then this little innermost thing is the nucleolus, and that's going to be like our DNA. So we'll get into this a bit more, but then, to kind of break down the major tissue types in the body, you can boil it down into 4 types. Epithelial tissue, which we're going to start with, then there's connective tissue, and then there's also muscle and nervous tissue, which will be, I'm sure, the subject of future presentations. So, okay. Next slide. It's not working. Sorry, can't see my mouse. Not sure what's going on here.
Can you try escaping from the presentation and representing it?
Yeah, that's what I'm trying to do. Jeez. Oh, sorry about this, guys. I'm like totally frozen. Oh, there we go. All right, let's reshare. Okay, there we go. Now we're good. All right, so to get in more to common features of all epithelial tissue. So with epithelial tissue, like I said, they're going to sort of be lining the surface of a lot of different things in the body. So one thing is that you're going to see is that cells are tightly packed together, and the cell membranes, the outer portion of the cell, are going to be held together by certain protein connections. And what these protein connections do is they basically form these like tighter junctions in between each cell, and it's going to maintain a sheet-like structure, so that all the epithelial cells are kind of like continuous with one another. Another thing about epithelial tissue is that it lacks a blood supply. And so what that means is that basically when you look at it under a microscope, you're not going to see any blood vessels. And it means that it has to be close to another tissue that has a good blood supply so that you can still get nutrients there, even though you don't have a blood vessel there. Another thing it has is good nerve supply. And the reason that's important is because epithelial tissue, one of the functions is it's going to respond to changes in the internal or external environment. And then we've been talking a lot about this basement membrane, but all cells are going to rest on that. And that's going to sort of help keep them all together and form a good surface. So to get into what a basement membrane actually is, so basement membrane is essentially this layer that's made of collagen type of protein and then glycoproteins. So if we kind of look at a couple of these images here, I'm not going to do annotating because I don't want another technical difficulty. But if we look at this top image here, we see how there's these sort of continuous line of cells that all kind of look like they're standing up next to each other. And we can clearly see like where it goes from dark purple to light purple. And that essentially is going to be our basement membrane, not including the cells because the cells are the actual epithelium, but essentially this sort of line that we can sort of draw is going to be our basement membrane. So the function of that basement membrane is it's going to provide structural support. So it's going to anchor all those epithelial cells to it and then to the connective tissue, which we're going to get into also. But that's the stuff that's sort of in between. And it's protecting against mechanical stress and just forms a barrier. Also can be a permeable barrier. So that's essentially to facilitate movement of a bunch of different things, nutrients, allergens, electrolytes, things like that, between connective tissue, which we said, between epithelial tissue, which we said has no blood supply, and connective tissue, which is under the epithelium and does have a blood supply. And then the last thing it's going to do is just form a physical barrier. And that's also preventing certain other substances and, for example, mutated cells like cancer cells from spreading to surrounding tissues. So it's really sort of a functional way to make sure that our tissue is functioning as it should. So to go into different types of epithelium, there's a couple of ways to break this down. So really 2 ways. We're going to break it down into the number of layers of cells that are involved in the epithelial layer, and then the shape of the cells. So really when it comes down to number of layers, there's two words we can use that we're going to use to describe our epithelium. So one is going to be simple. So if we have simple epithelium, it's going to be a single cell layer. And what that means with a single cell layer, it means that all of the epithelial cells attach to the basement membrane. And then stratified is the opposite of simple. So we have multiple cell layers. That's 2 or more cell layers. And Basically, what that means is not all of the cells are going to attach to the basement membrane. Only the bottommost layer is going to attach. And the layers on top of that are basically just going to anchor to the cells below it. So that's how we're going to sort of classify the number of layers that the epithelium has. We also want to describe the cell shape. So there's a few different ways we can describe it. And this is just sort of terminology to remember. But first one is called squamous. And what squamous means is sort of flat horizontal cells. So if we sort of look at the top of this diagram here, we see that these cells are really flat, compact, and look mostly horizontal. Then we have cuboidal. I like that name because I think it describes it very well. They look like cubes. Basically, you know, if we're looking at this in 2 dimensions, which we will be on slides, they basically look like squares. Oh, there we go. Okay, so yeah, that's going to be your cuboidal. And then columnar. So these are going to look like columns. They're basically going to be tall cells and look a lot more like columns, which is what we see sort of at the bottom here. So if our epithelium is stratified, this is just another thing in terms of naming. We're going to name it and we're going to classify it in terms of the top layer of the epithelium that we're going to see. So for example, if you would see, you know, some kind of epithelium where the cells that are attached to the basement membrane look more like cubes, but the top layer is more columnar, you would call that a columnar epithelium. It would be stratified as well, stratified columnar, because there's multiple layers. So a couple more oddball types of epithelium that don't really fit in well to this classification of simple versus stratified. We have pseudostratified, and emphasis on the pseudo here because it's not actually stratified. And the reason that we call it that is because we still have a simple epithelium, so to speak, in that there is only one layer of cells and all of the cells are attaching to that basement membrane. But when we look at the histology, when we look at it under the microscope with staining, the nuclei of the cells are not really going to be in line. So if we look at some of these other simpler stratified epithelium, we see that all of the nuclei are really like well in line with each other. Pseudostratified is not going to be like that. And then last one here is transitional epithelium. And this is kind of its own category because it's pretty specialized to one area of the body. You find it in the bladder and parts of the urethra. And it's good for there because it can sort of stretch and expand. And if we sort of look at this model of transitional epithelium, it's kind of like a bunch of, they're not really cuboidal cells. They sort of look like that, but they're not really. And they're kind of like the nuclei are all kind of clustered together, and there's several layers of cells that can sort of expand or contract and change the number of cells that you sort of see on top of one another. So in terms of correlating locations and functions of different types of epithelium, we'll start with the simple epithelia. So with this first one up top here, we have simple squamous epithelium, right? We have single layer of cells, of flat cells, so simple and squamous. These are very good for diffusion. That's really what we're looking at here. And so some of the things where, some of the places we might see simple squamous epithelium is places like the air sacs of the lungs. So we want diffusion of oxygen. And so that's not going to be able to diffuse over a very, long distance. So we want flat cells that are good for that. Then we have blood vessels. So especially when we're talking about very small vessels like capillaries, which are like, for example, bringing oxygen to the tissues around the body, we want things to be able to diffuse across that epithelium and that basement membrane very easily. So that's where you're going to see simple squamous. Simple cuboidal, we're thinking all about secretion. And so a lot of places where we're going to see this is going to be in ducts. And any place where we're really secreting different substances. And so where we might see this is in the kidney, for example, and other places where we're secreting things like saliva and stuff like that. And it's going to be for secretion and absorption. Next type, so simple columnar epithelium. So one thing that we're going to talk about a little bit more later when we get into what's on the apical surface of these cells, some specializations, is what we might see a lot with simple columnar and pseudostratified columnar is these little cilia things. And the reason that's important is because we're going to often see simple columnar cells in places where we want things to get moving and where we have some absorption and secretion. So a great example of that is going to be like in our digestive tract. And then pseudostratified columnar epithelium, kind of similar to simple columnar in terms of the places we find it. But a big thing here is that these are going to be good for secreting mucus and moving mucus around as well. So we see that a lot in the upper respiratory tract and certain parts of the lung. So now to go to the function of our stratified epithelium. So like we kind of see here, we have the same classification, just multiple cell layers. So here's multiple flat cell layers. That's going to be stratified squamous. And stratified squamous is really good for protecting against abrasion. So the main place that we see stratified squamous is going to be our skin. So our skin all over the body, that's going to be made up of stratified squamous epithelium with a bunch of protein that's sort of stacked on top of it. We also might see it in places that are really well exposed to the outside environment, things like the esophagus, the inside of the mouth, and then reproductive organs. So stratified cuboidal and stratified columnar, both of these are not seen in a whole lot of places, but really what we're thinking about is similar function to our simple cuboidal and simple columnar epithelium. It's just essentially larger and provides more protection. So for example, stratified cuboidal in larger glands like our salivary glands or sweat glands, we're going to see that additional cell layer could be more than two. And same thing with stratified columnar. We might see that in parts of the urethra and then the ducts of some of those larger glands. And then like I sort of talked about before, to kind of round it out with the transitional epithelium, that is going to be highly specialized and line the bladder and certain parts of the urethra that are closest to the bladder. And those sort of allow for more stretching, unlike any of the other epithelial types. So I sort of alluded to this earlier, talking about surface specializations, but really we're going to talk about three. So first one we're going to talk about is cilia. You guys may or may not have heard of these before, but essentially what cilia are these little hair-like projections that are on the apical or top layer or top surface of the epithelium. So if we see our basement membrane on one side, the cilia are going to be on the opposite side. The unique thing about cilia, relative to the other surface specializations we're going to talk about today, is that they can do active motion, which means they're actively moving and sort of sweeping things along, or in whatever lumen or compartment we're talking about that the epithelium is lining. So the cell type we're going to see these most on is going to be our columnar cells. So if we see simple columnar or pseudostratified columnar epithelium, that is in a lot of ways going to have, in most cases, going to have cilia. Microvilli, a couple of differences from cilia. So the difference here is emphasis on the micro because they are going to be a lot smaller. So if we look at these pictures here on the right side of the slide here, if we look at the apical surface of the epithelium up top, we can sort of see these like hair-like projections and things sticking out. The microvilli down here, it looks like sort of a very continuous line. You almost can't see it. You have to look really closely. But it sort of looks like it's just all one like little faded line, but it is very small projections. The function of that, because we don't have active motion like we do with cilia, it's more like passive. And so the main function here is going to be increasing a surface area for absorption of nutrients and other things like that. And so places where we'll find that is, for example, the intestines or the kidney, wherever we're trying to reabsorb things. And sometimes you might see this terminology, but microvilli are also sometimes called a brush border, in that they are very small, kind of looks like a little brush. And last surface specialization that we're going to talk about is something called stereocilia. This, again, you're going to see in way fewer places than cilia or microvilli. But similar to microvilli, these are passive motion, but they're just way larger. So they're substantially larger and really found in pretty much two places. The first one is going to be the inner ear. And so there's these hair-like projections that are on these certain receptors in the ear. And essentially what they do is as they move back and forth with the fluid within the ear, that's going to communicate sound waves. And that's sort of how our hearing works. So that's one place we're going to see it. The other place is going to be in the reproductive system, specifically in a structure called the epididymis. Don't necessarily worry about that, but it's a place where sperm are stored prior to ejaculation. And they're sort of just kind of like these long hair-like projections, much bigger than cilia, that are sort of assisting with absorbing fluid and transporting the sperm in that organ. So here we have a little bit of a quiz here. So unfortunately, I didn't do the poll thing, but if you guys want to type in your answers in the chat, that would be great. So we'll start with this one in the top left. So if you guys could take a guess, or just to sort of assess the understanding, what type of epithelium would this be in this top left image? So give it like a few more seconds here. All right. So I'm seeing some good guesses here. So yes, I'm seeing a few people for pseudostratified. That would be correct. So the reason that this is pseudostratified and not simple columnar, which I'm seeing, I guess, a couple other people put in here, but we see here, we can kind of see this basement membrane on one side. And if we look at the nuclei, which the nuclei are these really dark staining, like oval shaped things. And if we see that, those are not really in line with one another, but it's not really stratified because there's only one layer of cells. And so that's going to be why that would be pseudostratified. So how about this one to the right of that, the top right image? All right, I'm seeing some good stuff here. Very nice. This would be simple cuboidal. So like I said, it's in the name. These are, all right, maybe they're a little circular or oval, but that's all, tomato, tomato. This is simple cuboidal because we have these we see the nuclei in the center and kind of looks like a donut just around it. And we have a single layer of cells and we see that basement membrane that's sort of connecting them all together. All right, so how about this one? Well, I ruined it. All right. Anyway, so this one down here, this is going to be transitional epithelium. And the reason that we have it that way is because we sort of see this outer epithelium, we see our basement membrane here, and then our nuclei are kind of all scattered throughout. There's not really any rhyme or reason to the pattern here. But, kind of these several cell layers, and it doesn't really fit into an accurate description of any other type. That's transitional. All right, so then how about our bottom right one here? I won't click this time. Right, just give it a couple more seconds, but we are looking good here. Yes, this is stratified squamous epithelium. So kind of look pretty soft, and we have these, we see how there's like a whole lot of layers that are above this basement membrane. That's going to be stratified squamous. Very good. All right, so moving on here, last bit of epithelium talk is we're going to talk about the different types of glands. So really we can break this down into two things. We can break it down into endocrine glands and exocrine glands. So to start with endocrine, essentially what these are, it's a collection of epithelial cells and they're secreting hormones, and those are going to go directly into the bloodstream or extracellular space. Big difference here, you'll notice that they are conveniently highlighted in yellow, but endocrine glands, there will be no ducts. So these glands are just going straight into whatever lumen that they're supplying into. This is going to include all glands that produce hormones. That's our endocrine system. Anything that's circulating in the blood. Great example of this is going to be these cluster of cells called pancreatic islets. Those are going to be in our pancreas, and those are going to secrete a very important hormone called insulin. And that's going to, have a lot of control over our blood sugar throughout the day. So that's endocrine glands. Now to talk a little bit more about exocrine glands. Those are going to consist of a secretory portion, which often what we'll see is simple cuboidal epithelial cells. So we talked about the importance of that for secretion. With these glands, it's usually going to be simple cuboidal. Not always, but pretty much. And the difference here with exocrine glands is that we do have ducts that are transporting whatever material these cells are secreting and putting them into extracellular space or into the blood. And a lot of times, something else to think about in terms of exocrine glands, when we think exo, we're thinking outside. So a lot of times these are going to secrete into an area that's connected to the outside environment. So that's going to include skin, which would be, for example, sweat glands, mouth. We have saliva, that's going to be salivary glands, and then GI tract, because that's sort of protected from the rest of the body. And we're going to secrete a lot of different hormones, enzymes into there. So that is really it on exocrine versus endocrine.
Andrew, can I just interject just for a minute? It's Dr.
course.
Peterson. There's a
Yep.
really good question from Emily in the chat, and she's asking, and I think everybody it would just be easier to just talk about this than to type a really long answer. So she's asking, what's the difference in functionality in having no ducts and having ducts? So can I give it a try, and then you want
Yeah,
to
of course.
jump on?
Absolutely.
So with endocrine glands where I have no ducts, if I'm putting, as Andrew said, a hormone like insulin, it's going to circulate throughout the entire body. So it circulates systemically. And that may be a really good thing because in the case of insulin, we need it to go to almost every cell in the body. Where when we're talking about exocrine glands, we have ducts, and that takes us to a very specific location. And typically that is not something that we want to circulate systemically. So we talked about exocrine glands like salivary glands. We don't want saliva circulating throughout our whole body. We just want it to go to our mouth, to our oral cavity. But Andrew, jump in. Please fill.
That was, I could not have said it any better. That was, yeah, exactly. So really endocrine, full body hormonal systems that are going to affect a whole lot of different tissues. Exocrine glands, we're really targeting one specific area. All right, and so that's going to finish it up for epithelium. Now we'll move on to connective tissue. So here are the learning objectives for connective tissue. So we're going to talk about, first off, the different types, and we're going to break down what loose versus dense connective tissue actually means. And then the extracellular matrix, which we sort of saw in maybe a couple of pictures that we've looked at already. But we're going to talk a little bit more about the composition of that and its role in connective tissue and where we might find it. And then the last one, describing the basic components of loose connective tissue specifically, and a few different cell types and different types of proteins that we might see within that type of tissue. So general connective tissue overview. So what is connective tissue? So connective tissue is type of tissue that essentially is found right under that basement membrane or that basement surface of the epithelium. And what it's going to be doing is supporting and connecting different tissue types to one another, emphasis on the connective tissue. And it's basically, we're connecting muscles to organs to other things like that. And unlike our epithelium, this is going to have a very good blood supply. So it's going to be highly vascularized. So major components of connective tissue, we're going to have widely spaced cells. So unlike epithelium, where all of those cells were connected sort of in a line or by those tight cell-cell junctions, this is going to have like floating cells kind of going all around. And the other component of that is what is called the extracellular matrix. And that's going to consist of these protein fibers, which we're going to look at in a bit, and then some stuff called ground substance. And there's really three types of connective tissue in the body. So what we're going to focus on today is what's called connective tissue proper. And there's three types of connective tissue proper: loose, or sometimes called areolar connective tissue. And then there's dense irregular and dense regular connective tissue. Some of these other tissues are also considered connective tissue, for example, bone or adipose, fatty tissue, also blood or your lymphatics, but that will be subject for a later date. So to talk a little bit more about connective tissue proper, so to talk about the cells that we might find in here. So there's really two ways that we can classify the cells that we find in connective tissue, and that's Are they resident cells or are they transient cells? Now, resident cells, the name kind of helps because resident, what that means is that they are residents within that connective tissue. They are going to stay there pretty much regardless. They're planted in the connective tissue. And the main cell types that might consist of are cell type called fibroblasts. And fibroblasts are going to produce those fibers and ground substance, which we'll get into at the end of this slide. A certain type of immune cell called macrophages. And then we might see some adipose tissue, which also is known as fat. So that's our resident cells. Then our transient cells are the cells that have access and can get into the connective tissue when it needs to happen, but do not stay there. So they might be floating around in the blood. And then, for example, when we're talking about our leukocytes or white blood cells, in response to, say you get a cut on your finger or something like that, you're going to have those cells come in to the connective tissue and essentially attack any bacteria or any other pathogens that might have entered. And then once they perform their function of, basically providing an immune response in that area, they're going to leave and they're not going to stay in connective tissue in that area. So those are going to be the cells we find. Then we have different types of fibers. So fibers are going to be made of protein. One that you guys might have heard of already is collagen. And collagen, what we're going to see under the microscope is long, straight fibers, and these have very good tensile strength. And what that means essentially is that they're very good for being resistant to stretching. So they can kind of sustain any sort of abrasion or damage. Next type of fiber we're going to find is elastin fibers. I like this name because you can kind of think of it in terms of elastic, but it's made of a protein called elastin, and these are good for stretching and compressing. And so when they stretch, they're good because they can return to their original shape. And then the last type of fiber that we're going to find is something called reticular fibers. Now, reticular fibers are also made of collagen in terms of the protein structure, but when we look at them under the microscope, they're going to be more narrow and they're going to be a little bit more disorganized, and they're going to be sort of in this branching network. And so the way I kind of like to think about reticular fibers is it sort of forms like a filter or a net. That's sort of what they're going to look like. And so as such, they're found in organs where there's a lot of filtration going on. So for example, the liver, which is filtering out a lot of toxins and breaking down, certain drugs in your body. We're going to want a lot of filtration, so we might see a lot of reticular fibers there in the connective tissue. So last, to just touch on what this ground substance is. So like I was kind of saying with, a little bit less organization than we were talking about with the epithelium, there's going to be a lot of fluid in connective tissue, this extracellular matrix. And so ground substance essentially encompasses all the fluid that's in between. So ground substance, a couple of characteristics about it. It's pretty viscous. It's a transparent solution, and it contains a lot of different things, carbohydrates and proteins, and the amount of ground substance that we see is going to vary depending on the different type of connective tissue that we're looking at. So to start it off, we're going to start out with loose connective tissue. So we have a good sort of example of that on the right here. This is a type of connective tissue found all over the body. It's between a lot of organs binding them together, and it's just beneath the epithelial cell layer. And this is going to be very good for, again, diffusion of things like electrolytes, water, other nutrients like that. So if we look at this photo on the right, ground substance is all this stuff in between where we can't see anything. It's basically just the background color because that's just going to be like a viscous fluid where things are moving around. The fibers are going to be these branching, all these things that are sort of organized in a different way. So we can kind of look at, I like how these are labeled. These collagen fibers are going to be a little bit thicker, and they're going to be good for that tensile strength because they're a bit more sturdy. Then you have these elastin fibers, which are a lot thinner, and those are good for stretching. And we sort of see some of our resident cells here, but we see a fibroblast labeled. And fibroblasts, again, just to reiterate, those are going to be synthesizing all these fibers and all these proteins that we're seeing in here, as well as a lot of the ground substance. And again, we're not actually seeing any transient cells here, but lymphocytes and other white blood cells would be what we see, sort of what I talked about before. So that's our loose connective tissue. So now to get into our dense irregular connective tissue. So with this, we have fibers that are densely packed there is going to be less ground substance. So if we look at this picture on the right here, we're not seeing as much of that just transparent open area as we were before. So basically the reason that we're seeing less of that ground substance is because there's a greater amount of collagen fibers. Fibers are still going in random directions though. So that's why we call this irregular connective tissue, because the fibers are sort of going all over the place and can form this mesh-like network as well. Differences between this dense irregular and loose connective tissue is going to be that it's a bit more durable than loose connective tissue because it has more collagen fibers. We're going to be a bit more resistant to stretching, and we can withstand a bit more damage. And so a good example of this is going to be the dermis of the skin. So the dermis of the skin is essentially there's like 3 layers of the skin. The middle layer is something called the dermis, and that really helps give our skin a good protection against cuts and scrapes and abrasions that we might encounter in everyday life. All right, so now to get into dense regular connective tissue. This is our last tissue type. And if we look at this picture here, this is, we can kind of see the difference in the appearance between this and dense irregular connective tissue. Instead of those fibers going in all different directions, they're basically all aligned. They're parallel to one another, so they're all regular. The difference here, it's going to look very dense, very little ground substance is going to be present here. Notice how it's all lighting up pink, and we're seeing very little bits of like white spots. That's going to be our ground substance, but there's not much of it. We have a lower presence of resident cells. So to go back to loose connective tissue, because there's so much space, a lot of times we're going to see like more cells within that space. There's more room to move. But with this dense connective tissue, we're not going to see as much of that. So these have the greatest amount of tensile strength and resistance to stretch because of the amount and density of the protein fibers. A lot of times we're going to see elastic fibers with this. And so a good example of this connective tissue is tendons and ligaments. So we want to make sure that our musculoskeletal anatomy is sturdy and stable. And so we're going to need some very dense connective tissue to sort of resist any damage or stretching or anything like that that we might find. All right, so I believe this is the last slide here, but we'll do the same thing as we did before. And we'll take a look at, if you guys could put your answers in the chat, we'll start with this image on the far left. What type of connective tissue are we looking at? All right, fantastic. Everybody's got it. This is loose areolar connective tissue. We see more cells, we see fibers in random directions, a lot of ground substance. All right, how about this one on the right up top here? Perfect. Yes, this is going to be dense irregular. So by process of elimination and in the interest of time, I'm just going to click on this last one, which is dense regular. And I think it'll be good for comparing between the two. But we see how these fibers in the dense regular are very much aligned. And then up top here, they're sort of going in random in different directions. And then with the dense regular, we have less ground substance. And those are some references, and that concludes the presentation. So now we got some time for questions.
Thank you, Andrew. Excellent presentation. To preserve time here, to give Zach enough time for his presentation, we will take questions in the chat. And that way we can transition to our last presenter and give him enough time. Again, excellent presentation. Thank you very much. Okay. Well, last but not least, we're going to look at embryology. And we have Zach Mattiola, who is from Collegeville, Pennsylvania, and graduated from, I come from Ohio, and we call it the team up north. You all know it as the University of Michigan. And Zach graduated in 2022 with a degree in neuroscience. He is currently a second-year medical student at Drexel University College of Medicine. In his free time, he likes to watch sports, especially Michigan football, the Eagles, and the Sixers. He also likes to work out and play basketball and pickleball. Academically, he is interested in pharmacology, anatomy, and physiology. He wants to specialize in anesthesiology, critical care medicine, or surgery. So without further ado, I will hand this off to Zach to bring us on home for this evening.
All right. Thank you, Dr. Frank. Good evening, everyone. Thanks for bearing with us till the last presentation. I know it's a lot of information, but just got one more to go here. All right. So I'm going to be talking about basically an introduction to embryology, giving you some general terminology, talking about gametogenesis, fertilization, and implantation. And all of those words will make sense to you by the end of this, I promise. All right. So here are my lecture objectives. These are just taken word for word from the learning objectives on the Anato-Bee website, and I highly recommend you take a look at those and try and go through them and answer them because a lot of the questions are going to come directly from these learning objectives, okay? And I'm going to be talking through all of these in my presentation, but just try and go when you have some free time to go through these learning objectives and see if you can take a crack at answering them. All right, so we're going to jump right in. All right, so first I'm going to talk about the difference between embryonic/fetal age and gestational age. So this is essentially how we measure the age of a pregnancy. All right, so the embryonic and fetal age measures the age of a pregnancy from fertilization. All right, and fertilization is when the sperm meets the egg, they combine to form a zygote, and that goes and develops into the baby. All right, so the embryonic fetal age measures a pregnancy from the time of fertilization. As a doctor in clinical practice, that's not very practical. Okay, so we've developed a different way to measure pregnancy, and that's called the gestational age. And that's calculated from the last menstrual period. So if you have a pregnant patient that you're seeing in clinic and her last menstrual period was 3 months ago, she's 3 months pregnant using the gestational age. And I think this is a really good chart that outlines these different measurements. So the gestational age, you basically add on 2 weeks from the fetal age, okay? And we know that the last menstrual period will occur 2 weeks before fertilization occurs, approximately, okay? So it's a good kind of quick and dirty way to get the age of a pregnant patient, okay?
Yeah,
Okay,
can I just
so
interject?
sure.
Can I just interject? Just go back to that slide just for a moment, just so that they understand that ovulation happens approximately 2 weeks after the first
Yeah.
day of the last known menstrual period, and the viability of the egg cell once it's ovulated is about 24 hours. Hence, to make this simple, we say that ovulation and fertilization are virtually the same point in time. Just for clarification.
Exactly. Thank you, DP. Okay, so now we're moving on to the stages of embryonic development. We break these stages down into 3 phases, okay? The first is called the germinal period, all right? So this is the first 2 weeks of embryonic fetal age. So from fertilization to 2 weeks past that, that's the germinal period. And during this time, you get implantation. So the embryo is going to implant into the uterine wall. And that occurs in the first 2 weeks after fertilization takes place. The next phase is the embryonic period, which is weeks 3 to 8 after fertilization takes place. And this is a real important stage during the development of a fetus, okay? This is where the initial development of all of the organs takes place, okay? So you go from having no organs to beginning to start developing your brain, your heart, your intestines. Everything begins during this phase, and that's why it's so critical and important. Okay, and then the fetal period is week 9 to birth. During this period, you just have growth and further maturation of all the organ systems. Okay, so the fetus is going to grow. All of those organ systems that began developing during the embryonic period are going to continue growing and continue maturing until birth. Okay. All right. So now, I'm sure you've all heard that women who are pregnant should not smoke cigarettes, should not drink alcohol. There are certain foods they have to avoid. And maybe you've asked yourself why that is. Well, alcohol and tobacco are not good for a baby's development, okay? We call things like alcohol and tobacco teratogens, all right? Teratogens is just any substance that's going to interfere with the development of a fetus, okay? And it's especially important to avoid teratogens during that embryonic period, weeks 3 to 8. Okay, because that's when you're laying the foundations for all of the organ systems in your body. Okay, so if you introduce something that's going to interrupt with that initial development, those organs aren't going to develop correctly at all. Okay, so if you look at this chart, you see in blue here is the time of greatest vulnerability for each of these organ systems. And you can see that all of them have their time of greatest vulnerability during this week 3 to 8. Okay, so it's really important that you avoid some of these harmful substances during pregnancy. Okay, so now I just have a quick question for you if we can get a poll launched. So how is this chart measuring the fetus's age? Is it gestational age or embryonic age? All right, doing pretty well so far. Maybe give it a couple more seconds here.
I'll go ahead and close the poll in 5, 4, 3, 2, 1.
All right. Yeah, so good job, guys. So this is measuring the embryonic fetal age. So one of the key things here is you're looking at week 1. You have you have an embryo at week 1. If you are measuring using the gestational age, remember you don't have an embryo until 2 weeks after the start of the gestational age if you're using that to measure the pregnancy. And it also only lasts 38 weeks. Okay, I think I forgot to mention this, but gestational age lasts 40 weeks and the embryonic age lasts 38 weeks. Okay, so that's the way you can tell the difference here. All right, good. All right, so now I'm going to talk to you about mitosis and meiosis. All right, so we're going to start with mitosis. And the goal of mitosis is for one parent cell to divide into 2 identical daughter cells. Okay, it starts with interphase. And during interphase, the cell wants to grow. It wants to make sure it's ready to divide. It wants to make sure that its DNA is replicated so it can divide into 2 equal daughter cells. And it wants to make sure that the environment it's in is adequate to support more cells. Okay, so all of these conditions need to be met for division for mitosis to occur, and that happens during interphase. Okay, and I just want to bring this up because I know this is a point of confusion for a lot of students. After DNA replication occurs, you have one chromosome here that's composed of 2 identical sister chromatids, and they are connected by a structure called a centromere. You don't worry about that, but these are going to split apart during mitosis and go into each of the daughter cells. Okay, so this is one chromosome composed of 2 identical sister chromatids. Okay, so to start mitosis, we start with prophase. So we'll have the condensation of the chromosomes so that they're easy to move around. You'll have dissolution. The nuclear envelope will dissolve. Okay, and then you have the centrosomes, which are basically factories for microtubules. Okay, remember the microtubules are the structures during mitosis that are going to grab onto and pull the chromosomes. So these are made at the centrosomes, and these centrosomes are going to split to opposite ends of the cells during prophase. Okay, so that's prophase. Then we go to prometaphase. During prometaphase, the microtubules are going to be reaching out. They're going to be grabbing onto the chromosomes. They're going to be pulling them in opposite directions. And eventually, we're going to get to metaphase, where the chromosomes line up at the middle of the cell. This is called the metaphase plate, this point where they line up. All right. And then we go to anaphase. These sister chromatids are going to separate, and then the microtubules are going to pull them to opposite ends of the cell. And then finally, we have telophase and cytokinesis. During this stage, you have the nuclear envelope is going to reform, and the cytoplasm is going to be divided. All right. And then that's the end of mitosis. And at the end, you're left with two daughter cells, remember? Two daughter cells that are identical to the parent cell. Okay. Now we get into meiosis. Meiosis is a little more complicated. It's split up into two stages, meiosis 1 and meiosis 2. I'm going to cover them separately. And the goal of meiosis is for a diploid cell to divide into 4 haploid cells. Okay. Remember, human cells, almost every cell in your body has 46 chromosomes. It has 1 chromosome, 2 pairs of the 23 chromosomes that we have, 1 pair from your mom and 1 pair from your dad. Haploid cells have half that number of chromosomes. They have 23 chromosomes. And the goal of meiosis is to create 4 haploid daughter cells. Okay. And just to go over, I'm going to be using this term homologous chromosome a little bit. So just to define it for you. So like I said, you have 2 copies of each chromosome, 1 from your mom, 1 from your dad. Together, those are homologous chromosomes. Okay. They're not identical. They have the same genes, but they're not identical. So they're homologous. All right. So like with mitosis, we have interphase where the cell is going to grow and the DNA is going to be replicated. You're going to have those sister chromatids are going to be connected to one another. And then we go into prophase. Similarly to mitosis, you have dissolution of the nuclear envelope. You're going to have those centrosomes are going to migrate to opposite ends of the cells. But what's different here is those homologous chromosomes are going to come together and they're going to exchange genetic material with each other in a process called crossing over. Okay. And crossing over is really important because it increases genetic diversity in the daughter cells. Okay. So if you look over here, you see this chromosome has a little bit of red on it. This chromosome has a little bit of blue on it. And that's because they swapped those portions of the chromosome. Okay, and that's what crossing over is. And that occurs during prophase 1 of meiosis. Okay, and then we go to metaphase. Similarly, they line up, the chromosomes are lined up along the metaphase plate, the middle of the cell here, by those microtubules. But what's different between metaphase 1 of meiosis and metaphase of mitosis is that these chromosomes are going to line up with next to their homologous pair. So in mitosis, remember, they just line up end to end along the metaphase plate. But in mitosis, they line up next to their homologous chromosomes. Okay, and then we go to anaphase 1. And this is where the homologous chromosomes are separated to either side of the cell. In mitosis, it was the sister chromatids that were separated. But in meiosis, it's the homologous chromosomes, okay? And then finally, we get to telophase. You have the telophase and cytokinesis is when the cytoplasm is going to divide. And the major difference here is that at the end of telophase 1, you are left with 2 haploid cells, each with 23 chromosomes, okay? So by the end of meiosis 1, you already have haploid cells, okay? Whereas at the end of telophase and mitosis, you are left with 2 diploid cells, okay? All right. Oops, sorry. Now meiosis 2 is a lot more similar to mitosis than meiosis 1 is, but it's important to keep in mind that this is occurring in haploid cells with 23 chromosomes, not diploid cells. So same story you've heard before. Prophase, centrosomes are going to migrate to opposite ends of the cells. The nuclear envelope is already dissolved from meiosis 1, so we don't need to do that again. Metaphase, they line up along the metaphase plate. Unlike in meiosis 1, meiosis 2, the chromosomes just line up end to end. Okay. And then in anaphase 2, the sister chromatids are going to separate, just like in mitosis. Okay. And then telophase 2, same story. You're going to divide the cytoplasm. You're going to reform the nuclear membrane. And at the end, you're going to be left with 1, 2, 3, 4 haploid daughter cells. Okay. All right. Now, I have another poll question for you. So what is the primary difference between anaphase 1 of meiosis and anaphase of mitosis? So take a second here if I can get a poll launched. All right, good job, guys.
I'm gonna go ahead and close the poll in 5, 4, 3, 2, 1.
All right, great job. So you're absolutely correct. The right answer is C. So meiosis anaphase 1 separates homologous chromosomes. All right. Mitosis anaphase separates sister chromatids. Okay, so I think it's really important to go over each step of meiosis and mitosis and identify what's different between those two processes. Okay, so great job. All right, so why is meiosis important? Okay, well, meiosis is the basis for a process called gametogenesis. Okay, and gametogenesis, simply put, is the production of sperm in males and the production of eggs or ovum in females. Okay, and sperm and ova are called gametes, gametes. Okay, so I'm going to just quickly go over these two processes. I think the big thing I want you to take away from here is, where is mitosis important in this process and where is meiosis important in this process? Okay, so with spermatogenesis, you start with a spermatogonium. Okay, and this is like the sperm precursor cell. This guy is going to be hanging out and he's just going to be doing mitosis and producing the first cell in meiosis for spermatogenesis. He's going to be producing this primary spermatocyte. So he's just going to be hanging out, dividing and producing these primary spermatocytes. Once you produce these primary spermatocytes, then you can start doing meiosis. So this primary spermatocyte will do meiosis 1 and form 2 secondary spermatocytes. These guys will do meiosis 2 and form 4 spermatids. And then these spermatids, all they have to do is a little more maturation, a little more change a little bit, and you have 4 mature sperm from 1 primary spermatocyte. Okay? So mitosis is important for starting the process, kicking it off, and then meiosis is basically how the sperm get made. Okay, and I think it's important to know that males make sperm continuously from puberty until death. They're constantly making sperm until they die. All right, so now for oogenesis, similar story. Okay, you have an oogonium that's going to hang around and do mitosis and form those primary primary oocytes in this case. Okay, and then the primary oocytes can go into meiosis. The one thing that's different from oogenesis and spermatogenesis is that primary oocytes, after each meiotic division, one of the daughter cells is going to get all of the cytoplasm. Okay, so after the first meiotic division, this daughter cell gets all of the cytoplasm. And this other daughter cell, we call it a polar body. It gets no cytoplasm. It's just discarded. Okay, it's not going to be used for anything. And then we have the second meiotic division to give you the mature ovum. Again, all of the cytoplasm is transferred to one of the daughter cells, and the other cell is discarded as a polar body. All right. And unlike in males, females only make a limited number of eggs. Okay. They only make about 300 to 5,000 eggs, and they only ovulate about 400 to 500 eggs in their lifetime. Okay. And unlike in males who are just making sperm constantly from puberty until death.
Zach, I'm just interjecting. So this is on the YouTube recording. So just go ahead and again, reinforce that it's 300,000 to 500,000.
Oh, did I say 300?
Okay.
Sorry.
Okay.
It's yeah, so they make 300,000 to 500,000 total eggs, but only 400 to 500 are ovulated. Okay. So each time a woman has her period, he or she is ovulating an egg. Okay. And that can only occur, it's not going to occur 300,000 times in a woman's life, right? So only 400 to 500 eggs are ovulated. Okay. All right, so now fertilization. We've made sperm, we've made eggs. Now we're going to have them meet to create an embryo. All right, so fertilization typically takes place in the ampulla of the fallopian tube. So this is a uterus, okay? This is called the vagina, and this is the fallopian tube, okay? So a sperm is going to swim up through this uterus into the fallopian tube and hopefully encounter an egg when it gets to this portion called the ampulla, which is where fertilization occurs. From the female perspective, the female is going to make her eggs in the ovary. Okay, this is similar to the testicle of a male. This is where the egg cells are going to be made. And after the egg cell is made, when it is ovulated, it's going to be sent up into the fallopian tube. Okay. And if it gets sent up into the fallopian tube and it meets a sperm at the ampulla, then we may have fertilization occur. Okay, so the sperm meets the egg. What happens then? Okay, so the sperm has to get through a couple layers surrounding the egg first. Okay, the first layer is called the corona radiata. Okay, and the corona radiata is basically a thick layer of cells that exists to provide nourishment to the egg cell. Okay, so you see all these little pale dots. These are supposed to represent cells in the corona radiata. So the sperm makes its way through the corona radiata, and then it encounters a really hard roadblock called the zona pellucida, which is just a thick layer of jelly, basically, which the sperm can't get through on its own. So what does it do? When it encounters the zona pellucida, it's going to release a bunch of enzymes that chop up the zona pellucida to help it get through. And after it's done chopping up the zona pellucida, it's going to swim through and it's going to encounter the egg membrane. And when it encounters the egg membrane, it's going to fuse with the egg membrane and release its DNA into the egg. Okay? After that happens, okay, the sperm is going to, or the egg is going to basically harden the outer layer here to prevent other sperm from getting into the egg. Okay? So the sperm releases its DNA into the egg. Then the sperm and egg DNA are going to fuse to form a diploid zygote. Okay? So we have two haploid gametes, sperm and egg, fuse, their DNA comes together to form a diploid zygote with 46 chromosomes. Okay? Now, what happens next? Okay, so we've formed the diploid zygote in the ampulla of the fallopian tube. It's going to travel down the fallopian tube. All the while, it's going to be dividing really rapidly. So you go from 2 cells to 4 cells to 8 cells to 16 cells really quickly, okay? You eventually form a morula, which is basically a solid cluster of cells. I think it's 32 cells or something, if I remember right. And then this morula is going to take in a bunch of fluid. It's going to take in a bunch of fluid and form this fluid-filled cavity, okay? And at this point, we form the blastocyst. Okay, and the blastocyst is composed of 2 important cell populations, the outer cell mass and the inner cell mass. Okay, the inner cell mass is going to become the fetus, and the outer cell mass becomes the placenta. Okay, and then this is going to implant into the uterine wall, and we're off to the races. The baby's gonna, or the fetus is gonna start growing and maturing, and the pregnancy is gonna start taking place. Okay, what happens if it implants somewhere else? Okay, let's say it implants into the fallopian tube. Okay, this is called an ectopic pregnancy. Okay, if you've ever heard that term before. Okay, ectopic pregnancies occur when the cells implant somewhere other than the uterus, and then this fetus can start growing in the fallopian tube, and that can cause real major health consequences to the person carrying this pregnancy. Okay, all right, now the last thing I'm going to talk to you about are stem cells. Okay, so to give you so stem cells will differentiate into all of the major cells in your body. Okay, what is cellular differentiation? It's a process by which immature cells, like a zygote, this is a very immature cell. It's the process by which immature cells become specialized to perform specific functions. So differentiation is like when cells of the inner cell mass become a brain cell or a muscle cell or a heart cell, okay? That's cellular differentiation. So the zygote starts as totipotent, okay? This means that these cells can become any cell in the body, and they can also form the placenta, okay? That's what totipotent means. They can form anything. But totipotency is quickly lost when you reach the blastocyst stage. When you get to the blastocyst stage, cells of the inner cell mass will become pluripotent, okay? This means that the cells within the inner cell mass can become any cell in the body, but they cannot form the placenta, okay? Now, what is this business with primary germ layers? Okay, well, the cells in the inner cell mass will form what are called the three primary germ layers, the endoderm, mesoderm, and ectoderm, which you will hear more about. That's just basically, these three different layers will basically become different parts of the body. Okay, that's all that means. Endoderm, endo, think inside, these are going to become cells like within your gastrointestinal tract, basically cells that line the inside of your body. Ectoderm, ecto, think outside. These cells are going to become cells of your skin, basically things that line the outside of your body. Okay, you're going to hear more about this in a later session, so don't get too caught up on this now. The most important thing I want you to take away from this is that the blastocyst inner cell mass is pluripotent, and that means it can become any cell in the body, but it cannot form the placenta, okay? Only the outer cell mass can form the placenta, okay? And then finally, multipotency. Multipotency, these are specialized cells, okay? So they can't form every cell in the body. They can only form a certain subset of cells, okay? So for example, mesenchymal cells. You guys heard about connective tissue in the previous lecture. These mesenchymal cells are stem cells that will form cells composing connective tissue. So for example, bones, blood vessels, and muscle, okay? But they can't become a heart cell. They can't become a brain cell. They can't become a cell in your stomach. They can only form cells that make up connective tissue, okay? That's what multipotency is. They're just more specialized, all right? And that's, okay, I got a question for you here, okay. So all of the following tissues can be formed from cells of the inner cell mass except what? All right, good.
I'll end the poll since we're doing good on participation in 5, 4, 3, 2, 1.
All right, so the correct answer is D. But I get it. This is not a gimme question here. Okay. The key word here that I wanted you to focus on was placenta. Okay, remember the inner cell mass is pluripotent. It can become any cell in the body, blood vessels in the femur, neurons in the brain, photoreceptors in the eye, sure, but it cannot form any part of the placenta. Okay, that's the outer cell mass. This will form the placenta. Okay, not the inner cell mass. All right. All right, and that is all I have. So if you have any questions, feel
Jack,
free to ask them.
there was one in the chat, and how about if you give it a shot and I will jump in, or one of the other faculty will, if you need some help with it. So
Okay.
do you see that what happens in the case of identical twins regarding fertilization? You and I knew that this would probably come up. So
All
do you
right.
want to?
That's a great question. So hold on. Let's go back to fertilization. Identical twins. Isn't it just when no, I can't answer it. I'm sorry. I have no idea.
I know you can. It's just late in the day. So for those of you tuning in, and for those in real time, and those of you that are going to watch this, I think the simplest answer for now is that you can have at the level of totipotency where we're moving towards the morula. So Zach, maybe you could use your cursor. As long as the cells are all totipotent, because they're dividing through mitosis, you can actually have a complete separation of a cluster of two different clusters of cells that then travel down the fallopian tube, and both will then become implanted, and they can both develop into an embryo and fetus. They'll generate their own placentas, etc. So they'll have their own blood supply. The longer in time that division of clusters of cells takes, meaning if it happens day 4 or 5 after fertilization, then that can still proceed normally. But what can happen if it happens right at the time of implantation, because those cells have started to specialize, it becomes more likely that the identical twins will be conjoined. So it's really all about, rather than having cells dividing their cytoplasm but staying within a single mass, they've sort of lost the connections and they form two masses of cells. It's a little bit more complicated than that, but maybe for tonight, that's enough for you to process all of that.
I think that was beautiful, Dr. Peterson, and I'm just going to give you an even more simple version, a Cliff Note version. The sperm fertilizes the egg, so it's one sperm fertilizing one egg, and that egg can further divide itself. And I agree with Dr. Peterson, they can actually have two placentas. There are cases where they can share a placenta. And then there's the case, if all of you are wondering, well, if that's identical, what's fraternal? Fraternal is when someone can produce multiple eggs. So instead of ovulating one egg, they can produce or ovulate more than one. And then a sperm can fertilize each, and they would each implant at different locations within the uterus. If you were wondering, because there's identical and fraternal. And I'll be more than happy to share, I do have fraternal twins. So I'm very familiar with that process, and they're one now and they're amazing. So we have a question.
Yeah, I have a few, but about addressing the one that you just said, is it one sperm that's fertilizing two separate eggs, or is it two sperms, like to each, that are being implanted in different places?
So for the fraternal or for the identical?
Fraternal.
So for fraternal, it is two eggs ovulated, and it's two separate sperm that fertilize the egg. Yeah.
And I was also wondering if the outer cell mass also forms the umbilical cord as well as the placenta.
Joanne, you are asking such great questions, and the shortest answer to that question is yes, but it's a little bit more complicated
Yeah.
than that. But great question. And so I'm just going to say you're exactly correct. There's just more detail that we'll explain as we move forward towards the local competition in January.
Okay, thank you.
And I'd just like to lastly thank all of our presenters this evening, Hailey
Oh.
Long, Andrew Briskin, and Zach Mattiola. And I'm going to hand it back to Dr. Haggerty for final
I think there's one
comments.
more question, Dr. Frank. Sorry.
Let's just because we're on time, I'm just gonna remind everybody that and if anybody's willing, we can stay back for at least 5 more minutes, but to close the session out, because it is 8 o'clock. Thank you all for coming for session. We have some links in the chat. You can do our survey. We would greatly appreciate it. And then you can also and maybe as I'm talking, Mary, if you can go to the very top of it and copy and repaste the links. So there's a survey link that you can take for us. We would greatly appreciate it. And there's also the registration link. So if you haven't registered yet, please do so. And then a friendly reminder that our next tutoring session will be held on Sunday, September 29th. Okay. I think we can stay back and answer the last question here, but I did want to make sure that we ended at 8.
Thank you, everyone. Thank you, high school students, for
Thank you.
joining us, and a big round of applause for our second year
Yes.
students. Great job.
Thank
all
you
of
very much. Thank you, all of you. Yes.